Is your friend coming?
I'm
I
not
think,
sure.
okay. I think let me wait for about a minute and then we can start. Okay, since the lesson is being recorded. It's okay. I will grab a message in the WeChat. Yeah. Okay, let me start the lesson. Yeah. Okay, I think last week we stopped until this part. Okay, so I guess that you already know that the plant tissues they are exist in three conditions, which is from the turgids, which is the plants they absorb a lot of water inside and store in their vacuole, and flaccid means that the plant cell they lost a very large amount of water. Oh, your friend is coming already. Hi, Eden. Hey. Hi. Hi, Eden.
Hi.
Okay. Sure. Okay. So I will continue. For the last week, yeah, we stopped until plant cell. Okay. So you already know that in the plant cell, they are exist in three kind of conditions, which is the plant cell, they get a lot of waters and then they will store waters inside in their vacuole. So they will become a turgid plant cell. A turgid plant cell means that inside the plant cell, they are filled with a large amount of waters. Then how about the flaccid? A flaccid and the plasmolytes, they refer to the plant cell, they lost a large amount of waters. So until their vacuole will be shrinking in their sides. Okay. So in a very severe conditions of losing water by the plant cell, it will cause the cell membrane pull away from the cell wall. So it will create a space between the cell membrane and the cell wall. Just appear in this way. This way. I think you can see my screen, right? I'm sharing it. Let me confirm. Yeah, I'm sharing the screen. So today we learn about animal tissue. See, this is the animal tissue. Okay, the example of animal tissue where we learn for today is about red blood cell. Okay, so red blood cell is very interesting. Okay, it's found in our body. So red blood cell is classified as animal cell or plant cell. Which one? And then Suyang, red blood cell is classified as animal cell or plant cell.
Animal cell.
Yes, correct. It's animal cell. This is because that red blood cell, they don't have cell wall. They only have cell membrane. You can see? Yeah, they only have cell membrane. The structure of the red blood cell is something like this. Yeah, with the cell membrane. Okay, like this. Oh, my drawing is not too nice. Okay, yeah. So the shape of the red blood cell, they are known as the biconcave disc shape. Do you heard about this term before? This biconcave this DISC shape. And then please always remember that red blood cell, they don't have nucleus. Yeah, they don't have nucleus. It's very interesting, right? They don't have nucleus. Do you know why red blood cell, they don't have nucleus? It's because that there's a reason. Because they want to increase the total surface area to transport more oxygen to body cell. So this is the reason that red blood cell, they shouldn't have the nucleus. Okay, so for your information, during the development of the red blood cell, initially the red blood cell, they are developed from our bone marrow, right? So after that, they will mature, they will grow, and then they will mature. So in the maturing process of the red blood cell, they will remove their nucleus away. Okay, so since the red blood cell, they don't have the cell wall, they only have cell membrane. So they are quite fragile compared with the plant cell. So how fragile is it? So let me go through with you. So in animal tissues, animal cell, they also just similar to the plant cell. They can gain the waters and then they can lose the waters. So since the animal cell, they don't have the cell wall. See, they don't have the cell wall. So they will cause the effect, which is the rupture of the whole cell if there is an excess amount of water enters into the animal cell. Yeah. So when you place the animal cell in a very strong sugar solutions, that this will cause the, maybe I can draw in this way. You have three kinds of solution, right? Hypertonic, isotonic, and the hypotonic. So imagine that if you place the red blood cell, into the hypertonic solutions, the hypertonic solution refers to a solution with a very high concentration of the molecule. For example, a very highly concentrated salt solution. Yeah, this, okay, very high concentration of the salt. Okay, concentrated. Concentrated means that there are very less amount of water, right? And there are very higher amount of the sodium chloride ions. So this will cause that the red blood cell are being placed in the hypertonic solutions. So the red blood cell itself, they will become hypotonic. Okay, they will become hypotonic compared to their surrounding highly concentrated salt solution. So the red blood cell itself, they will become hypertonic. Red blood cell will become hypotonic. Since the surrounding solution is hypertonic, it's very highly concentrated sodium chloride solution, so red blood cell itself becomes hypotonic. Hypotonic means that the water potential is low. Hypotonic with the high water potential, okay? High water potential. Hypotonic means that the content itself contains a very higher amount of water. So hypotonic means there is a high water potential. Okay, and then the solution itself is hypertonic. That means they are low water potential. So the golden rule for the osmosis is the water will diffuse from the red blood cell with a high water potential into and enter into the surrounding solution with a hypertonic, which is they have a low water potential. So water diffuse out from the red blood cell. So this will cause the red blood cell appear in this shape. See? This shape. So this is meant by the crenation of the red blood cell. These conditions, which is the red blood cell, losing their waters, and then they will appear in this star-like shape. So they know as a crenation of the red blood cell. Or sometimes they will refer as a crenated red blood cell. Then how about when you put the red blood cell in the isotonic solution? Isotonic means that they have a very, they have the same water potential. So how many water enter into the red blood cell? It will be how many of water will leave, leaving the red blood cell. So the net movement of water is equal to 0. So there will be 0 net movement of water. This means that how many water molecule diffuse inside? Maybe about 10 water molecule. If there's 10 water molecule diffused from the surrounding water solutions inside the red blood cell, there will be also 10 water molecule leaving from the red blood cell to the surrounding solution. So they cancel the effect of each other. So there is a movement of water. However, there's about 0 net movement of water because they cancel each other. So this is isotonic. So in the isotonic solutions, the red blood cell, they will still remain in their shape. See, their shape is they doesn't face any changing. Okay, red blood cell remain their shape. Then how about when you place the red blood cell into the hypotonic solutions? What happens? Hypotonic solutions means this. They have a higher amount of the water molecule compared to the salt solution, for example. So hypotonic means that you are placing the red blood cell in the solution which is very high water potential. So when you put the red blood cell in a very high water potential, so the red blood cell itself, they will appear become low water potential compared to the solution surrounding it. Low water potential. Okay, so this is cause the water, how the water will flow. This will cause the water diffuse from the hypotonic solutions and it will enter into the red blood cell. Enter. So the red blood cell, they will gain a very large amount of water and then they will increase in size. However, red blood cell, they will gain large amount of water and increase in size. However, they do not have the cell wall, so they are unable to resist high pressure and it will burst. Because there's too much water flow inside, creating a very high pressure. So since the red blood cell, they don't have the cell wall to support them, only the very fragile cell membrane. So this will cause the whole red blood cell to burst. So this bursting of the red blood cell, they are known as hemolysis. Bursting of the red blood cell. Hemolysis. Hemo means blood. Okay, just like hemoglobin, the proteins found in the red blood cell, hemoglobin. So hemolysis, lysis means bursting of the red blood cell. So you can have a look, okay, how the red blood cell is being burst. So this is very short video. I show you how the red blood cell is being burst because the water flow inside. Hemolysis. Okay. I think the internet is not in good connection now. Can you see the YouTube video? You can see, right? Ethan, Suyang?
Yeah.
You see. This is red blood cell. See. Yeah, they are waiting to be burst because you're placing them in very hypotonic solution. See, red blood cell, they gain a very large amount of water, right? So you can see, wow, they are like expanding in their size. And then at the end, what happened? You see how they burst. See, they are going to burst now. You see this? See. Okay, hemolysis. Okay. Oh. Okay, so this is a statement that you should know, okay, and then should be included in your answer. Hypertonic solutions. Okay, red blood cell itself, they have the high water potential compared to the hypertonic solutions. So the water will be moving out. So the cause the cell will be shrivel. Shrivel means reduced in the size of the cell. Okay. Isotonic means the cell, they don't have any change because there is no net movement water. No net means the movement water, they will be cancelled each other. Okay. how many water move inside the cell will be how many water move outside the cell. Then how about hypotonic solutions? Okay, hypotonic solution, it will cause the water move from the outside solutions into the red blood cell. So the red blood cell will gain the water and then they will swell up. See, swell up. Swelling up means that increase in size of the red blood cell, and then they will lyse. Lyse means burst. Yeah, so this is the red blood cell. Okay, it's very straightforward. Okay, then how about the strip? This strip that I refer to the potato strip. So there'll be, they like to come out these potato strips experiments in your paper 4 or paper 6 exam, so you can see what will happen. Okay, these potato strip refer to the plant cell. So that means that they have the cell wall. This is a potato strip. You like to eat potato, right? During when you go to visit McDonald's. So when you place the, oh my god, why the video is still moving? Let me remove away the video. Oh shit, what happened? Okay. Hey. Okay. When you remove the, when you put the potato strips into the distilled water, so what will happen? Okay, imagine that potato strip, they are comprised in something like this. They have something like skin. Okay, we call it as epidermal layer. Skin. Epidermal layer. So these epidermal layers, remember that it doesn't able to increase in their size for the epidermal layer. Because this epidermal layer is very hard. Okay, it's a hard layer. So another side, it will be the flesh of the potato flesh. So only the potato strap, the potato flesh, able to absorb the water, they will be causing increase in size for the potato strips. So when you place the potato strip into the distilled water means that there is only presence of the water molecule without any solid. So it's classified as a hypertonic solution. Okay, hypotonic solution. So the hypotonic solutions, they will be causing the water diffuse from the hypo solution enter inside the potato strip. They will enter inside. Water molecule will enter inside. So what will happen? So initially, we cause the potato strips, initially they remain in this way, right? See? Which is the both epidermal layer and the both flesh layer, they are in the same size. But however, since the water diffuse inside, so the flesh layer, the flesh part of the potato strips, they will be absorbed in water. So when they absorb in water, they will cause them to curve in this way. See? So this side, there will be increase in their length. This side will be increased in their length. So they will be curving outwards. So it curves outward, see? Curving outward with the epidermal layer on the inside. This is because that the potato flesh part absorb large amount of water. So they're able to curve outwards. So when you place the potato strips into the isotonic solutions, maybe it's like 70 sucrose solutions, so it will cause the both epidermal layers and the both the flesh parts. Okay, the whole length will remain in the same shape and also the same length. But when you put the potato strips into the 30% of sucrose solutions, that means it's a very hypertonic solution, a very, very high amount of the sucrose molecule inside it. So it will cause the potato strip itself become hypotonic. See? Okay, itself become hypotonic. So they will be losing water outside. Okay, there will be water diffused from the potato strips to the outside the external solutions. So when the potato strip lose the water, this will cause the flesh reduce in their length. So they will appear in this way. Something like this. See? See, they are curving inwards. curving inwards. This is a flesh of the potato, flesh. And then this is the epidermal layer. Okay, so remember that the direction of the potato strips curve either outwards or curve inwards. If they absorb the water, they'll be curving outwards. Okay, because the curving parts they are referred to the flesh part of the potato. This one, flesh part. The flesh part means that the part that we eat. Okay, yeah. So that's it for the potato strips. You just need to know their appearance. Okay, how they are curving outwards or curving inwards based on the type of solution that you put inside. Okay, so let me go to. Okay, and then when you write the answer for the osmosis, then please write that the water enters into the cell by osmosis. So they will cause the cell become turgid. Yeah, turgid in the plant cell. Okay, so when the water leaves the cell by osmosis, this will cause the potato cells, the potato strips to plasmolyze and become flaccid. Yeah, so this is a term that you should include in your answer. Okay, so we finished about the osmosis, and then now we talk about the active transport. So what is meant by active transport? Active transport, you should know about the, we call as the description for the active transport. Active transport, that means is moving of the particles against, see? You see from the low concentrations to the high concentration means that they are moving against the concentration gradients, moving against concentration gradient. Since they are moving against, that means that they are moving from the low concentrations to the high concentration, right? So you imagine that you're just like climbing a tree, not climbing a tree, climbing a mountain. You're climbing from the lower side, and then you reach a peak to the upper side. So in this process, you will require a lot of the energy input, right? So you can climb from the low side to the very top side. Yeah. So remember that active transport, they occurs in two conditions only, which is you must have the ATP energy. You must have this, have this energy, ATP energy. And then secondly, you must have the carrier proteins. Together with these two conditions, with the concentrated gradients, active transport able to occur. Okay, so please remember about the definition, which is the movement of particle against the gradient with the help of carrier protein and also the ATP energy. And then this ATP energy or energy, they are being produced from the cellular aerobic respiration. This one, cellular aerobic respiration. They are able to perform this active transport. Okay, I hope you still remember the equation for the cellular aerobic respiration, which is initially they will use the glucose and the oxygen. This one, C6H12O6 plus the oxygens, and then they will release the carbon dioxide, water, and also they will release the energy. And then please remember to write the balance equation with 6, 6, and 6. Yeah, so this is equation, the balanced chemical equation for the cellular aerobic respiration. Okay, I hope you understand. Okay, in exam, they like to ask about the applications. For example, active transport are being used in what kind of the conditions? They are being used in the process which is they require the energy input. For example, uptake of glucose. This one, uptake of glucose by the epithelial cell in the villi of the small intestine, and also the kidney tubule in the nephrons. So this one you will learn about in the next few chapters. So I just want to know that after that you eat the food, right? So the food will be digested by the various kind of enzyme. So for the fruits, okay, for the foods which is from the carbohydrates, for example, you eat the rice, okay, you eat the rice, carbohydrates. Rice contains carbohydrates, right? It contains the starch, actually, starch. So after you eat the food, so the food will be digested. They will be undergo the digestion by the enzyme digestion. So after they undergo digestion is a process which is they will break down a large molecule of the food particle into very small molecule of food particle. So they will become, at the end, glucose. They will become this glucose. Okay, so glucose is the simplest form of the nutrients molecule, simplest form. And glucose is being readily absorbed by the villi of the intestine. Readily being absorbed by the intestine villi. Yay. Okay. So this is how you're able to get your nutrients for your cell. So you see, when the glucose they will enter into intestine villi. Intestine villi, they will be in this structure, like this. Okay. So you can see that it's a very we call it highly folded villi, right? So this is the intestine cell. This we call as an intestine cell. Intestine cell refer to the villi. Then the outside we call as the intestine lumen. Okay, so once the glucose are being released in the intestine lumens, glucose are like this. Glucose, this is a glucose, right? Glucose is a 6-carbon molecule. So I put a 6-carbon compound here. So the glucose will be absorbed. See, they will be absorbed into the intestine villi. Because the intestine villi, they contain the blood capillary inside. It's something like this. Inside of the intestine villi, they contain this blood capillary, like this. This. So this will help to allow the glucose to be absorbed into the blood capillary, and then they will reach the bloodstream. And then after that, they will be processed by the liver, and then they will reach the body cell, because body cell need glucose for cellular aerobic respiration. You learned before, right? So how the glucose transport into the blood capillary in the intestine villi? So the answer is they transport it by active transport. Active transport. So why they are using active transport? This is because that in the lumen, okay, they have because a lower concentration of glucose. However, in the intestine cell, they contain a higher concentration of the glucose. So there will be active transport with the help of the carrier proteins and also the ATP energy. So this is the low glucose into the cell which is have the high glucose. Okay, so you should know that our intestine villi, they will absorb the glucose through the active transport. Okay. Yeah. This is one example. Then another example will be the kidney tubules in the nephron. I'm not sure you learned about this before or not. In the kidney tubule, there's a lot of parts, okay? So there's one part we call as the proximal convoluted tubule. This one, proximal convoluted tubule. So this proximal convoluted tubule, they will help to absorb the glucose, okay, the glucose from the kidney filtrates. So it's something like this. Maybe I can draw you a picture about the nephrons. So nephrons. What is nephron? Okay, nephron is a functional unit of the kidney. Okay, it's a functional unit of a kidney. So in one kidney, you contain billions of the nephrons. So it's functional unit of the kidney. So the nephrons, they are help to filter the substance. Okay, so let me draw you an example of nephrons. So nephron, they will be something like this. Something like this, right? And then they'll be like, okay, until they reach like this. Okay, sorry, my drawing is not too good, but I will show you that this is we call as the kidney tubule. Okay, we have about this part, and this part. Okay, then let me talk about this part. Because this part we call as a proximal convoluted tubule, which is we call as a PCT. Okay, I will draw as PCT. So during formation of the urine, the urine will be passing through from the blood capillary, and then they will reach this proximal convoluted tubule, right? Which is the urine. And then this proximal convoluted tubule, what they do, they will do the selective reabsorption. This one, selectively reabsorption. What is the meaning of reabsorption? Means that they will reabsorb some neutral molecule or water or any kind of particles from the urine filtrate enter into the bloodstream. Okay, from urine filtrate. Urine filtrate is found inside the the kidney tubule. Urine filtrate into the bloodstream. So in this process, there will be absorbed about 100% of the glucose, which is found in the urine filtrate, back into the bloodstream. 100% of glucose. So Ethan Suyang, do you think that in your urine, you're able to discover glucose? Yes or no? In your urine, you're able to discover glucose? Can or no? Do you think your urine contains glucose?
No.
Yes or no?
No.
No. Okay, no. Luckily, she'll say no. Yes, your urine shouldn't discover any glucose. If you have glucose in your urine, that means like, you have some problem with your health. For example, maybe you got diabetes. So the diabetes patient, they will have the glucose in their urine. Because in the normal healthy patient, in the normal healthy person like us, we shouldn't discover any single glucose in our urine, because our proximal convoluted tubule, they will 100% reabsorb all the glucose from the urine filtrate enter into bloodstream. Okay, so there will be reabsorbed 100% of the glucose. Will be reabsorbed by the PCT. Okay, from the urine filtrate into our bloodstream. Yes. So this process is by active transport. Okay. Apart from the glucose, okay, other molecule also like a large polar molecule, for example, amino acids. Okay, amino acid also being reabsorbed back into the bloodstream also by the active transport. Amino acid, for example. Okay, so apart from glucose, amino acids, you also have the iron, right? Iron, for example, is a sodium ion, potassium ions. Those also using the active transport. Okay, for example, we talk about root hair cell. Root hair cell is a very common topic to coming out in the exam. Root hair cell, did I show you a picture? Oh, I didn't show you a picture about root hair cell. I didn't show you a picture. It's okay. Maybe you can draw down, okay? This is a root hair cell, right? It's just like someone pointing a finger to you. So this root hair cell, they contain a very long extensions. This is root hair cell. Root hair cell. So the function of the root hair cell is they tend to absorb water and also the mineral iron, right, from the soil water. So this is soil. So root hair cell, how they absorb the water. So water will be entered into the root hair cell by osmosis. This is the water. Okay, they will enter by osmosis. This is because that the outside the soil, we call it soil water. Soil water itself, they contain a higher percentage of the water. So they have a higher water potential. Okay, compared to the root hair cell. Okay, root hair cell, we call it the cell set. This one, cell set of the root hair cell, they contain a lower water potential. So you can write in this, we kind of answer. Water will diffuse from high water potential. Okay, from the soil, which have high water potential, enter into cell set of root hair cell with the low water potential by osmosis. Yeah, please remember this term, osmosis. This is the water. Okay, so you know it is by osmosis. So osmosis means that it's a passive transport. It's a kind of passive transport, and it doesn't require any help of the ATP energy and also the carrier proteins, right? Yeah, so it's a passive transport. Just need water, just need a selective permeable membrane, so you're able to did the osmosis. Then how about how the root hair cell able to absorb the mineral ion, just like sodium or chloride? So how they absorb? They will absorb mineral ion, okay, sodium or chloride, also by active transport. Okay, this is because that outside the soil water, they have the lower percentage of the sodium chloride ions. But inside the cell set, they have the higher concentration of the sodium chloride ions. So you can write in this way. I think I don't have last page. You can write, I think I write here. Yeah, I write here. So the root hair cell, the water, not water, okay, sorry. The sodium ion, for example, sodium ion will actively transport from soil water with a low concentration of the ion, then enter into the cell set of the root hair cell with a higher concentration of ion. Okay, then you can add on with the help of the carrier proteins, because it's active transport, right? Carrier proteins and also ATP energy from cellular aerobic respiration. Aerobic respiration. Yes. Okay, so always remember that how root hair cell able to absorb water and mineral ion from the soil water. Water is by osmosis. Then mineral ion is by the active transport with the help of carrier proteins and also the ATP energy. This one. So when you write the answer, you will get full mark already. Okay, then now we talk about another example. See, sodium potassium pump. So what happened to this sodium potassium pump? Sodium potassium pump is a kind of process which is they are happen in a lot of cell. Okay, a lot of cell they have this sodium potassium pump. So the story is like this. This is a cell, right? Cell they contain inside. This is the inside of the cell. And then this is outside of the cell, which is referred to the extracellular fluid. Outside of cell. So please always remember that inside of the cell, they contain a very high amount of potassium ion. They contain a very high concentration of potassium ion. However, the outside of cell, they always have high concentration of sodium ion. Potassium ion inside the cell, outside higher sodium ion. So in the sodium potassium pump, what will happen is the cell with the carrier protein, carrier protein like this. Okay, what they do, they will tend to transport the sodium outside. See, 3 sodium outside. They will transport the sodium from inside of the cell to outside of the cell with a very high concentration of sodium ion. Then how about the potassium? Potassium, there will be transport into the cell from the outside. Okay, from outside into inside with 2 potassium being pumped inside. So this is called a sodium potassium pump. So every cell, they can did this sodium potassium pump, okay, which help to, they transport the sodium outside and then the potassium inside the cell. So you can see that there will be contribute to the very high concentration of potassium inside the cell and also very high concentration of sodium outside the cell. This is because of this sodium potassium pump. So you can see that sodium are being pumped from inside to outside because inside the cell, they have a high amount of potassium, but they have a very low amount of sodium. See, very low amount of sodium. Inside the cell, they have more potassium, very less sodium. However, your sodium potassium pump, they're able to pump out the sodium from the low concentration gradients to outside the cell, which is very high concentration of sodium. So that means it's from low concentration inside the cell to the high concentration outside the cell. So this is against the gradient. Maybe I can write this down. Sodium actively pumped from inside the cell with a low concentration of sodium. Okay, to outside the cell with a high concentration of sodium. See, so this means like against. So they are doing what? They are doing against the concentration gradient, just like you climbing a hill from the very low level to the top layer against the concentration gradient. So they require the ATP energy and carrier proteins. Okay, and so the carrier proteins. Then how about the potassium? Potassiums, okay, they are also actively pumped. They are actively pumped from outside the cell with a very low concentration of sodium or low concentration of potassium. Because outside the cell, they have more sodium, but they have lesser potassium. Okay, outside the cell, they have more sodium ion compared to the potassium ion. Inside the cell, they have more potassium ion compared to sodium ion. Just remember in this way. So when they pump from the outside of the cell with the low concentration of potassium into inside of cell with a very high concentration of potassium. So this is also similar, which is against the concentration gradient. Okay, so that's a story for the sodium potassium pump. Yeah, I hope you know that. Then we talk about the additive transport across the cell membrane is how additive transport works. So it actually works by using the carrier proteins. Okay, you require the carrier proteins. So these carrier proteins are usually found inside the plasma membranes. So they're able to help to pick up some specific molecule, Okay, either it's large molecule, large and polar molecule, so they're able to transport against the gradient. So they require the carrier proteins and also the ATP energy generated from the cellular aerobic respirations. Okay, why they need the ATP energy? This is because that sodium potassium pump it's a pump, right? Pump means that they involve in the changing in the size of the carrier protein. They will cause the carrier protein to change in their size, so they need the ATP energy. Okay, carrier protein slightly change in their size. Slightly change in the size. So they're able to pump the 3 sodium out and 2 potassium ion in. Okay, so generally those are the information for the energy transport. Okay, so yeah, the most important is you should understand the concept. Okay, and then you should know that which area will transport the ion to which area. Okay, then let's talk about the applications of the movement across the cell membrane. Yeah, so you know about you already learned about 3 ways, right? 3 ways, 3 methods of the how our plasma membrane able to transport the particle, which is about simple diffusion, osmosis, and active transport. Facilitated diffusion, you just no need to learn it up. Facilitated diffusion is not in your syllabus. In your syllabus, in your IGCSE syllabus, you only learn about the simple diffusion, osmosis, and the active transport. So you can see, simple diffusion is only involved in the small, simple molecule, for example, the gas exchange. Okay. And also any kind of gaseous exchange involved in the plant. Okay, you still remember that the carbon dioxide, they will be uptake by the stomata, and then they will reach the airspace in the mesophilic cell. So those are happen by simple diffusions. Then when the oxygens, okay, inhalations of the oxygen, and they will enter into trachea, and then they will reach the lung until the alveolus, and then they will enter to the blood capillary. This process also happen by the simple diffusion. It's very simple process. Okay, so whenever involved in the gaseous exchange, usually is occurred by the simple diffusion. Gaseous exchange means that they will be involved in the carbon dioxide and the oxygen transport of the carbon dioxide and the oxygens. Usually they will be involved in the simple diffusion. And then how about osmosis? Okay, osmosis is only involved in the water molecule, transfer of the water molecule only. Water molecule. So root hair cell, yeah, and also the reabsorption of water by the renal tubule in the kidney. Okay, you will learn this later, which is our kidney tubule able to reabsorb the water, okay, from the urine enter into the blood capillary. Then how about active transport? Okay, active transport is being used to actively absorb the glucose and amino acid in the intestine villi. This villi is from the intestine. And then absorption of mineral salt by the root hair cell, and then how to transport the sucrose from the leaf into the phloem tissue. Okay, this is for your information. Later you will learn that the difference between plant and human. Okay, in human we transport our nutrients majorly in form of glucose, right? I think you have already realized this, because we always talk about cellular aerobic respiration, which is they help to break down the glucose to release the energy. So in human, we transport our nutrients in form of the glucose. Then how about in plant? In plant, they will be transport their nutrients not in glucose but in the sucrose. Okay, they will transport in the form of the sucrose. Transport nutrients molecule in sucrose. Sucrose is a kind of the monosaccharide. Oh, it's not monosaccharide, it's a disaccharide, sorry. It's a disaccharide. I think you heard about disaccharide before, right? Oh, sorry. You heard about disaccharide before? Disaccharide means 双糖. It's a kind of the glucose. Okay, disaccharide. So plant, they will transport in form of sucrose. Please remember that. Okay. So that's it for the chapter 3. And then here's some question you can attempt later. So okay, now I'm going to discuss your homework did on the last week. So let me go through your homeworks. Okay. Thank you for submitting the homework. I can see that you have a great understanding on this subject. Okay. Oh, I'm scaling the screen, right? Can see my screen? Yeah, you can see my screen. Do you have any question to ask me, Honey? Ethan and Suyang, do you have any question to ask me? No? Yes?
No.
Okay. I'm not sure. Am I speaking too fast? Do you think am I speaking too fast?
No, not really.
Oh, okay. I really hope that. Okay. Yeah. So let's discuss about the homework last week. I'm not sure which one is did by Suyang or which one is did by Ethan's. I'm not sure, but just put your answer together for me to mark. Okay. So both of you get correct in the first questions, which is turgid. And then this also, okay, additive transport. So you already know that the additive transport is a process which is they will transport the molecule from low to high. This is low, this is high. Very easy to be identified. Low to high concentration. And then this one, okay, yes. And then this also, correct. Okay. Osmosis, okay, what is the condition for the osmosis to happen? Osmosis, first, they need to involve in the water molecule. Osmosis is only involved in transport of water molecule, right? But not other solid. And then they require the sedative permeable membrane. Sedative permeable membrane or partial permeable membrane. Okay, which is the membrane? This is the membrane, right? And then they have a very, very small hole. The hole is small enough to allow the water molecule to diffuse through the hole. Yeah. Okay, for this one. This one, the answer is donkey. Why? This is because that this is the dialysis tubing, right? This is tubing, dialysis tubing. Okay, you just imagine that this dialysis tubing, they are actually refer to the semi-permeable membrane. This is this semi-permeable membrane. So you put the sugar solution inside the inside this dialysis tubing, right? So surrounded with the water solution outside. And then you know that water is hypotonic, which is high water potential. Water itself is hypotonic with a high water potential. Then how about the sugar solution inside? The sugar solutions become hypertonics. Okay, they become hypertonics with a low water potential. So the water molecule able to diffuse from outside in and enter into the dialysis tubing, which is because they contain the sugar solution. So they will be moving like this, moving inside. So when they're moving inside, what will happen? When the water diffuse inside, the water level will be decreased outside. See, and then the glass tube, the level will be increased. Okay, so the level of the liquid in the glass tube will be going up. This is because that the outside water, they have the higher water potential compared to the sugar solution. So the answer is donkey. Okay, simple questions. Yeah. Okay, this is talking about the active transport. You require the carrier protein and also the energy, both. This is all correct. Okay, and then for this is the plant cell. Okay, then Ethan and Suyang, you tell me, how do you describe this condition of plant cell? Flaccid, turgid, or plasmolized? Which one? How do you describe this condition of plant cell? Flaccid, turgid, or plasmolized? Which one? Hello? See? your plant cell vacuole will be shrinking inside, right? And then what happened? Your cell membrane pull away from the cell wall. So it's a turgid cell or it's a flaccid plant cell? Which one? Hi. I'm waiting for your answer.
Flaccid.
Yes, it's flaccid. Correct. It's flaccid. Flaccid. It's not turgid. Definitely not. Okay. So yeah. So what made the plant cell appear in this way? It's because that this person, they will put this plant cell into the solution P, which is the solution P itself become since it's losing water, right? Losing water to the outside. That means that the plant itself is hypotonic, yeah, hypotonic. Okay, hypotonic means they have a very high water potential. High water potential. So that means that the outside solution P, they are hypertonic with a very low water potential. Okay, outside solution P, they have a very low water potential with a hypertonic. So the answer will be C, which is the water potential of solution P is lower compared to the inside of cell. So the cell become high water potential compared to outside, right? So that's why the water will be diffused from inside of cell, which is they have a high water potential, to outside of the solution P, which is very low water potential. So the plant cell, they lose the water and then they will become plasmolized or flaccid. So this is their term. And then, yeah, this is, for this one, the answer is C. Okay. Sugar pressure inside the plant cell varies when the plant cell are placed in the external solution of different concentrations. Okay, what is meant by sugar pressure? For example, when your plant cell, okay, when plant cell absorb large amount of water, and they will store water inside the vacuole. And then what happened? The vacuole take a lot of water, right? So vacuole will be increased inside. Okay, vacuole will expand. Vacuole will expand, okay, and increase inside. So Adam Suyang, let me ask you, for this particular plant cell, which is they get a lot of water, their sugar pressure will be increasing or decreasing? If you are the plant cell, you get a lot of water, your sugar pressure will be increasing or decreasing? Waiting for your answer. Increase or decrease?
Decrease.
Decrease. You say decrease. Okay. The answer will be increase. Increase sugar pressure. So remember that when you are the plant cell, you get a lot of water. Water coming into your body, right? So now your body is full with water, right? So you are building up the pressure. So this pressure, they refer sugar pressure. So if you get a lot of water inside, so that means that the water, they will be exert the pressure against the cell wall, right? So this will cause the sugar pressure. This will create the sugar pressure. So you look at this question, you can see that this sugar pressure inside the cell, what they do, wow, they are slowly decreasing until reach zero. See, decreasing. Okay, so what happened to the plant cell at the point X? You see the sugar pressure becomes zero already. Sugar pressure become zero. So that means that there's no pressure exists inside the plant cell. Why there's no pressure? Because you lost a large amount of water. Okay, you lost water from your body, you don't have the pressure inside your body because the water will create the sugar pressure inside your body. Can you get me? Adam Suyang? Can you understand?
Yeah.
Yeah, okay. So when you are losing water, actually you are losing the sugar pressure. So you become flaccid. Okay, well done. Yeah, I think other question you get the correct answer. Okay, now let me open a note for you. This one. I think I sent in the WeChat group. Let me check. Okay, you can open chapter 3, 学霸笔记, okay? Between Adam and Suyang, you can read about the Chinese character, right? 你们知道怎样看华语字对吗？华语字。Okay. Let me show you this. 你们看懂华语字对吗？
Yeah.
Okay. Because majority of my student is also from the China, okay? So I will write some Chinese character inside. Yeah. I think this note is very good for your understanding, because you can say that you learn about three type of the transport, right? Which is the simple diffusions, osmosis, and the active transport. Okay, so you can know that simple diffusion is the most simplest one because they only involve in the gaseous exchange. For example, oxygen and carbon dioxide in the alveolus, and also simple diffusion are found in the stomata when the carbon dioxide will be uptake into the stomata, right? They will enter into stomata and then they will enter inside the leaf and then they will perform the photosynthesis. So those process, which is the carbon dioxide enters into the leaf through the stomata, it also happen by the simple diffusions. Okay, so other than that, okay, small uncharged molecule when they are trying to pass through the plasma membrane also happen by simple diffusion. Okay, this one, the neurotransmitter transmit across synapse. Okay, this is another story. Okay, you will learn this in the chapter 14. Chapter 14. Okay. So, what is the condition for the simple diffusions? They do not need to require carrier protein. No carrier protein. They just require the pass through of the plasma membrane. See, they just directly pass through the plasma membrane, and then the continuous gradient is they will be moving from the high to low, which is down the continuous gradient. Okay, so they don't require any ATP energy. It's a kind of passive transport. Same with the osmosis. Osmosis is almost similar to the simple diffusion. Just osmosis, this osmosis, they only involve in the water movement only. So the definition for the osmosis is the water will always diffusion from the high water potential to the low water potential and then pass through this semi-permeable membrane by osmosis. So they will pass through this semi-permeable membrane by osmosis. So this is the definition for the osmosis. Okay, osmosis, they don't really require carrier proteins, no need. But they require what? They require the semi-permeable or partially permeable membrane. Okay, and then the golden rule, they will be always moving from the high water potential or hypotonic hypotonic solution to low water potential, which is the hypertonics. Down the continuous gradient, and then they also no need require ATP energy. And then let's study about the carrier protein. The active transport. So the active transport, they are involved in moving of the particles, which is the large polar particle. Okay, large and polar charged particle, such as sodium and A+, right, or potassium. Okay, or the chloride, and also other like the glucose and proteins or amino acids. Okay, amino acids also involved in the active transport. When they're trying to absorb the amino acids from the intestine villi, are also involved in the active transport. So active transport, they require the carrier proteins. So they require the membrane or not? Yes, they require the movement of the particles across the plasma membranes. This across means what? This across means that you want to travel maybe you want to travel a river, right? Yeah, this is a river. Okay, you want to cross the river to reach the opposite, the opposite place, right? Then you need to ride a boat with you. So you are crossing the plasma membrane. Need the help of the carrier proteins. Okay, so the concentration gradient is you are moving from the low concentration gradients to the high concentration gradients against the, yeah. So this is mean by against the concentration gradient. And then you require the ATP energy. So it's a kind of active transport. So sometimes the question, they will just give you the distilled water or pure water. This one, distilled water or pure water. This distilled or pure water, they refer to the high water potential without any solids. Means that they only contain water molecule inside a solution without any solid. So they are referring as a high water potential. And then, okay, red hair cell, you know what happened, okay, which is they will absorb water by osmosis and absorb the mineral iron by active transport. Okay, and then the osmosis, okay, you can read out by yourself. It's actually moving from the hypertonic to the hypertonic now, okay? Okay, then, for this one, turgid and the flaccid, okay. This like to be coming out in the exam, turgid and flaccid. So why the plant cell, they need the tubular pressure is because that the tubular pressure, they will help to support the plant so they're able to stand upright. Okay, because the plant cell, they don't have backbone like us. In human or in animal, we have a backbone. We have very strong rigid backbone to support it, right? So we can stand upright. But plant cell, they don't have it. Plant cell, they only able to absorb the water. So the water will create the tubular pressure for the plant cell, so they're able to stand upright. So you can see the turgid plant cell and the flaccid plant cell. So how is their appearance? Okay, you should know how to draw the turgid plant cell and the flaccid plant cell. Yeah, sometimes they will be coming out in the exam. So they ask you to draw a diagram. Yeah, so this is an example of picture that you can draw. Okay, in the turgid cell, you can see that vacuole increase in size. See, vacuole become very large central vacuole. It's so big until they press the nucleus to the periphery area. So a very big nucleus. Okay, you can draw a very big vacuole, and very small nucleus at the periphery area, and then some chloroplast inside. And then the most important part is you can you can help to draw the cell wall slightly bent outwards. See, this is a cell wall, right? Cell wall, they are slightly bending outwards. So this is turgid. Yeah. Sometimes you need to describe. Okay, you can describe a turgid plant cell is a vacuole expand increase in size. So you can create a very large central vacuole. So the cell wall are slightly bent outwards. Okay, this is because that cell membrane press on the cell wall, so they create a tubular pressure. Also, there's no space between the cell membrane and the cell wall. Then how about the flaccid? You can see the flaccid cell. Okay, flaccid cell, you can see a very vacuole, they are shrinking inside. So vacuole becomes smaller, shrink and reduce in size. This is because that vacuole lost the water. They lose their water. So they will cause the cell membrane pull away from the cell wall. So there's no tubular pressure. Reduce tubular pressure. Reduce tubular pressure. Okay, you should understand that how plant cell get the tubular pressure is when the vacuole, you can see that when the vacuole absorb the water, right, they will expand. So once they expand, they will cause the cell membranes, okay, to press on the cell wall. So this is the moment that which they're able to create a tubular pressure because the cell wall is being pressed by the cell membrane due to the vacuole increase in their size. Okay, so I hope you understand that. Yeah. Okay, I think that's all for this chapter. Yeah, I think that's all. Okay, for this gut cell, gut cell is not so common to be coming on exam, but some question they will ask you about how the gut cell able to take in the water or lose the water. So you should know that the gut cell, they able to open and close, right? The gut cell, yeah, the gut cell, they help to open and close the stomata. Since the stomata is being controlled by the opening and closing of the gut cell, okay? So the gut cell, they can become turgid or flaccid. Turgid gut cell means what? Turgid gut cell means that the gut cell inside the cell, okay, they have the hypertonic compared to the outside. See, inside hypertonic, outside become hypotonic. So they will cause the water diffuse in the gut cell. And then the gut cell, okay, you can see they do contain the corpus. Also, they contain the vacuole. They have a small vacuole inside the gut cell. So this small vacuole inside the gut cell, they're able to absorb the water too. They can get the water too. So when they get the water, they will expand in their size. So when they expand, the gut cell, they will be turgid. Yeah, they will become turgid. So when the gut cell becomes flaccid, it's because that the gut cell losing the water. The gut cell inside become hypotonic than outside, right? Because outside they contain a higher concentration of solutions, means that higher salt concentrations. So gut cell itself become the hypotonic. So when they become hypotonic, they will be cause the water losing to the outside. Okay, from inside to outside. So the gut cell, you can see the vacuole will be shrinking in their size. So overall, they will become flaccid. So when the gut cell becomes flaccid, stomata will close. Yeah. So when the gut cell become turgid, expand inside, they will cause the stomata to open. Okay, so the closing and opening of stomata is, they will help to control like how many coming outside to be intake into the plant cell, or also how many of the water molecules should be evaporate out through the stomata. Yeah, so that's all for the chapter 3. So I think we have some time, right? So maybe we can try to do some chapter 4 question. It's not chapter 4, paper 4. Yeah, we do some paper 4. Yeah. Okay. You can open your chapter 3, paper 4. Sorry, I'm not sharing the screen. Let me share the screen again. You can open the, I think it's this one, right? Sorry. Oh. Okay. You can open the document that I sent in the WeChat group, which is the chapter 3, paper 4, part 1. Let's try the part 1 first. This one. Okay, after you're ready, let me know. And then we can start to do the question together. Okay, have you opened it? Chapter 3, paper 4, part 1, this one. Yeah, part 1. Okay, you have opened it, right? Can we discuss?
Yeah.
Okay, thank you. Sometimes you can see this IGCSE exam, they like to come up fill in the blank, which is very easy for you. Please score it when they come on this kind of question. Water move into and out of cell by what term you should write. And then water is known as what? Because it can dissolve the solute. So you think that what term you should fill inside? The movement of water is caused by osmosis. Okay, so this is osmosis. Yeah. And then water is known as the solvent because it can dissolve solute. Solvent means that any solutions which is they contain the water molecule inside, then they can add it with another solute. For example, sodium chloride or glucose. So water is a universal solvent because it can allow many kind of solute to dissolve inside. Okay. So this is a leaf. What happened to this leaf cell or plant cell? Okay, you can see that. Yeah, the vacuole. You see, the vacuole appears smaller or larger inside. This is a vacuole, right? Vacuole is like here, this one, vacuole. So this vacuole increase in size or they are reducing in size, this vacuole. Increase or decrease? So see, they put this leaf cell into the solutions. So the water potential of solution is lower than the water potential of the content of the cell. So that means that the outside, right, they have a low water potential. low water potential means that they have a very hypertonic. They are hypertonic solutions. So this means that the leaf cell itself become high water potential, right? High water potential. So they are losing their water from inside to outside while water come out. Okay. So they want you to sketch the expected appearance of the cell after it had been in the pure water for the 3 hours. Okay. So now you can see your plant cell appear in this way. Okay. Before that, Ethan and Suyang, tell me, this plant cell is a turgid or flaccid plant cell? Turgid or flaccid?
Flaccid.
Yeah, correct. It's flaccid. Okay, it's flaccid because you can see that the vacuole will be stringy inside, right? And also, your cell membrane pull away from the cell wall. Oh my god, flaccid. Yes, it's flaccid. Or you can say plasmolized plant cell. Okay, now you put this flaccid plant cell into the pure water for about 3 hours. So what happened to your plant cell? You put this flaccid cell into the pure water, right? So what happened? So they ask you to draw. Okay, sketch means draw. So how you can draw for this plant cell? Okay, you know that pure water, right? Pure water is hypertonic. Hypertonic means that it's very high water potential. High water potential. So high water potential means that the water will be diffused from outside and enter into the plant cell. So plant cell will get the water and then they will appear become a turgid cell. So let us draw a turgid plant cell. Come. This is the turgid plant cell. Okay, how we can draw? Okay, you can draw something like this. A fat plant cell. Okay, fat plant cell. You can draw like this, and then cell membrane. And then you can draw a very large vacuole. So large vacuole. Yeah. I think it's not large enough. Maybe I can draw larger a bit. Very, very large. Okay, initially your vacuole is very small, right? Now you're going to draw a very large vacuole. See, this is vacuole. The vacuole is so large until they will press the nucleus to the periphery area. This is nucleus. Okay, this is nucleus. And then corpus. Okay, you have the corpus. How many you have? 1, 2, 3, 4, 5, 6, 7. Then you just draw it like this. Corpus. 1, 2, 3, 4, 5, 6, 7. Yeah. Corpus is not important. Just most important is that you should draw. Okay. This is the vacuole. Okay, this is the vacuole. Okay, then this is the cell wall. Then this is a cell membrane. And then, yeah, that's all. Okay, most important that you should write, you should draw like the cell wall slightly bent outwards. Okay, you can see, actually your cell wall is slightly bent outwards. And then vacuole large. A very large centralized vacuole, so big. And then you should write that there's no space. See, you can see any space between cell wall and cell membrane? No, right? No space between cell wall and cell membrane. No space. See, no space. You can see the flaccid cell, they have the space, which is here. This is the space. See, this is space. Yeah. This is the space between the cell wall and the cell membrane. The space. And then for your information, sometimes the question they will ask you, okay, Since the flaccid plant cell, they are immersed in this, what kind of solution name? They didn't say the solution name. Or they just state a solution. For example, yeah, the leaf cell will put, they are being put in the solution, for example, X. Okay, solution X. And then the plant cell becomes flaccid already and appear in this appearance, which is a flaccid plant cell. Then they will ask you, okay, in this space, they contain what kind of solution? Contain what kind of solution? In this space between the cell membrane and cell wall. So the answer will be solution X. They contain the solution X between the space. Why? Because that outside it will be surrounded by solution X, right? Yeah, this flaccid plant cell, they are outside, it's being immersed in the solution X. So if there's a space between the cell membrane and the cell wall, right? So this causes the solution X able to diffuse through the cell wall and enter into the space. So this space are being filled with the solution X. Yeah, this is if the question come out in this way, this is your answer. Okay, so why? I think you should know that cell membrane and the cell wall, the characteristic is, you can write it down. Cell wall, it's okay, I ask you. Ethan Suyang, tell me, cell wall is semi-permeable or is fully permeable? Which one?
Fully permeable.
Yeah, correct. Very good, you know the answer. Yes, but cell membrane is not like cell wall. Cell membrane is semi-permeable, or is called as selective permeable. Cell membrane is semi-permeable. Because the cell wall is fully permeable, so actually the cell wall, they contain a small hole. So they can allow the external solution just flow inside, enter into the space. But the solution won't able to enter into the cell membrane, because cell membrane is very particular. Cell membrane, you know, they are plasma membranes only permeable to certain type of the molecule or ion to flow inside. So that's the story. Okay, so this is the answer. You should write. You should draw in this way. Okay, then a plant was not watered for 1 week, so show a series of photographs of plant during the week. So you can see that how plant develop become in this way, which is the plant cell already wilt. Okay, see, use the term sugar pressure in your answer. Sugar pressure means that you should include sugar pressure in your answer. 3 marks. Every time when you answer the question, please remember to look on the mark. Okay, please don't write about 10 points for just 3 marks. Just if there's 3 marks, you just write about 4 points only. 4 points is enough for you. Sugar pressure, okay? So you can write that the plant will, after 7 days. Now, this is because that plant have undergo lack of sugar pressure. See, because they are losing the water, so they don't have sugar pressure to support them. Thus, plant unable to stand upright. The vacuum of plant cell lose water. So no sugar pressure. Okay. No sugar pressure push against the cell wall. And then that you can say that the mesophyll cell itself, mesophyll cell, are not providing support to plant cell. So losing water by the plant cell means plant cell become flaccid and plasmolized. Okay, the keyword is, you can say that let a tuber pressure, so lose water by the vacuum of plant. So there's no tuber pressure, help to push against the cell wall. Tuber pressure, okay? Then losing water, so they become flaccid and plasmolized. Yeah, you can get a full mark if you're able to include those terms in your answer. Okay, I hope you get it, yeah? So that's why you need to water your plant every day. If you didn't water your plant, they will become in this way, die and wilt. Okay, I go in next question. This one, I think this one, okay, maybe you can have a try. This show the villi in the small intestine, then state the name of the other part of small intestine. Okay, this you will learn later. This I can give you an answer. The answer will be duodenum. It's a part of the small intestine because small intestine are being divided into three parts. Right? Three part. There's three part in the small intestine. Okay, you will learn it in the chapter 7, human nutrition. Okay, you can see this is an intestine, right? So where is the blood capillary that I mentioned just now? The blood capillary are found in this way. This actually is a blood capillary. So they help to absorb the nutrients after digestion. And then for your informations, there is villi and also there is a microvilli. Microvilli are found on the surface of the villi. So it's something like this. This is a villi, right? Oh, sorry. This whole thing is a villi. This whole thing. Microvilli is found at here. Here, see? This is microvilli. It's much more smaller than villi at here. Microvilli. So both villi are to increase the surface area for the absorption, for the nutrient absorptions. See here, this is microvilli. Okay, so epithelial cell of the villi, they will absorb the nutrients by diffusion and transport. So tell the difference between the active transport and the diffusion. Okay, so when the question asking about this, describe how the active transport difference from diffusion. That means that your answer must only include in the active transport only. So you can see how I write the answer. Active transport require the ATP energy. Okay, to transport particle against the concentration gradient. Then active transport I think I should separate this term into two words. Sorry, this one. Okay, they require energy. Energy produced from cellular aerobic respiration. And then active transport help to transport particle against a concentration gradient from low concentration gradient to high concentration gradient. And then the third one, you can say that the eddy transport, they require the help of the carrier proteins, because this only involved in the eddy transport only, but not in the diffusions. Okay, require the help of the carrier proteins in plasma membrane. So 3 mark, 3 answer. ATP energy, okay, against the concentration gradients, carrier protein. That's all. So this is the answer that you can write. Simple and clear cut. And then for this one, microvilli. Are you still there? Sorry. I think, oh, okay, you're still there, right? Ethan, yeah. Okay, let me finish last question, okay, and then we can end the class. Microvilli, okay. You can see that microvilli are shown in the picture above, right? So the answer is they trying to increase the surface area. Surface area for the nutrient absorption. So the keyword is surface area, nutrient absorption. Maybe you can write to increase surface area to, so you're able to increase the, to increase, you can write to increase the rate for nutrient absorption. So 2 mark. Okay, so I think we'll stop until here. You can attempt in the next few questions for the question number 10 and then and 11. Yeah, you can attempt. So this is your homework for next week. Okay, so I hope you enjoyed the class for today. Then we'll meet in next week. Okay, bye.
Thank you.
Thank you, Ethan. Thank you, Suyang. Bye. Have a good rest.